in nature. That's much of what we want to do in science. We want to understand nature, but we'd also be interested in predicting the future in terms of biological systems, particularly humans, and also especially control, being able to understand how we can control systems in order to promote health and cure disease. So examples are determining genes and cell signaling pathways that cause breast cancer. This may help with facilitating, supporting precision medicine, finding targeted medications that affect those pathways and control the cancer or cure it. Discovering clinical effects of a new drug would be another example, and uncovering the mechanisms of pathogenicity of a recently mutated virus that's spreading rapidly in the population, another example of causal discovery. So the reason why we decided to pursue this center is two main reasons: the algorithmic advances that have been made in causal discovery and representation in the last 25 or 30 years, and secondly, the availability of big biomedical data, which, as we all know, is becoming much more rich and plentiful. So this is basically just recapitulated here, discovering networks from this data using these algorithms. In terms of the big data, there's a variety and richness of biomedical data that's becoming available. High-throughput molecular data, such as whole genome sequencing data, is an example of that. In the clinical arena, EMR data, and in the U.S., most physicians are using some form of an EMR. It's been quite accelerated in usage in the last 10 years. So that kind of data is becoming available for analysis to try to understand better some of the causal relationships. Population health, social media is providing a lot of sensors, a lot of possibilities for understanding what's going on outside the traditional healthcare system. And of course, these three can be integrated in various interesting ways. So a great and wonderful time to be involved in biomedical data and research, data analysis and research. So the basic idea is the appropriate analysis of these data has great potential to advance biomedical science. And what we're doing is combining these two to generate causal networks of various types to try to promote that science, and providing these networks to the biomedical scientists themselves. So the basic workflow then is illustrated schematically here, where we have prior knowledge, and there is a lot of prior knowledge, as we know, in biomedicine, so we want to have that represented in some form. We do have some data, as I just mentioned, becoming available, do a causal analysis to generate causal networks. And I should say the data is both observational data and experimental data. We know a lot about how to do causal analyses with experimental data. What's more challenging, but also increasingly more relevant, is being able to analyze observational data for causal relationships. And of course, we all know the mantra of correlation is not equal to causation, and that's certainly literally true. But what we've been exploring is maybe not as widely appreciated is if you have enough statistical dependencies among variables, sometimes the patterns of those statistical dependencies in very interesting ways suggest strongly causal relationships among a subset of those variables. So it's more nuanced than the aphorism about causality, correlation not equal causation. So this is what we've been exploring, and these causal networks embody the output of that analysis. The idea is that these networks will then help spur causal hypotheses, generation by biomedical scientists, finding parts of those networks that are novel, that contain causal relationships that have been inferred from the data, or at least purported causal relationships that are novel and interesting. And what we're trying to do is find those that are, as I said, novel, significant, and also valid. We would want the have a high, basically, predictive value or precision, that the arcs that we output in these networks actually, most of the time, are correct, so that they lead to, when you do follow-up experiments, those experiments tend to validate those findings, and in this way, try to help improve the efficiency of the cycle of hypothesis generation, discovery, and then re-experimentation to get additional data, and the circle basically continues with additional data and additional analyses. So let me give you an example of causal network discovery that actually was published quite a while ago, over 10 years ago in Science by Karen Sachs and others on protein signaling networks learned from multi-parameter single-cell data in human T cells. As I said, in Science, it was in 2005. So this is a diagram from her paper, their paper. It's a portion of a cell signaling network in human T cells. And on the top, you can see various membrane proteins, and what you have then is a cell signaling network on the inside of the cell below, and the orange indicates various, mostly proteins, signaling proteins. The arrows indicate essentially causal relationships among those signaling proteins, and you've got about 11 of those illustrated or diagrammed here. The other thing you see are the green little squiggly circles, which indicate interventions that they made in various parts of this network. And the green ones are activators of those signaling nodes, and the red ones from around 6 to 10 indicate inhibitors. So what they did is they went in and they used basically different interventions on these cells, and then they used a cell flow cytometry, and they used to basically look at these cells, and they used immunofluorescence, which was able to, in the experiments they did, show the levels of these proteins for about 11 of these proteins that you see here in the orange. And so each cell basically provided a sample in terms of the protein levels under various experimental interventions on these activators and inhibitors. The reason why they studied this network in large part was because it's well understood network, and they wanted to see if these methods could essentially confirm, reproduce the known network. So this is a example. This just sort of illustrates the overall experimental design, where here you had the perturbations that I just mentioned of various types that they tried. You had a flow cytometer. It basically was able to look at each of these cells. This is a single cell here. Are you able to see my pointer, Sean? Okay. I guess maybe Sean's on mute. I just wanted to make sure everybody can see what I'm pointing to here.
Yes, I am on mute, and yes, we can see your pointer.
Okay, thank you. I didn't want to do all our talk and be making a wrong assumption. Thank you. So anyway, okay, this is a single cell here, and so under some perturbation condition, and what you get is this readout here of the intensity of each of the proteins in the cell signaling network that they're measuring. And so you get that for this cell, for this second cell, you get another set of sort of measurements. And so they got a few thousand of these samples, effectively, that under different conditions, and they did a causal Bayesian network analysis using a Bayesian method that had been developed that could take both observational data as well as experimental, as well as, in this case, it's actually a mixture, because you do interventions on some nodes and you observe the others. So, and that's oftentimes, you know, what we have in experimentation. And then they developed a causal Bayesian network and looked to what extent was it correct relative to consensus knowledge in biology. So the bottom line is that the expected Of the 17 that they output, 15 are very well known. 2 of them, the green ones here, were less well published. There was like maybe a paper on them, but it was tentative. So what they did was, but it was suggested by the algorithm. So what they did is they did wet bench experiments, which indeed did validate these as being causal relationships here between these proteins or phospholipids, and in this case proteins. So there was one that was reversed. So in that sense, if you think that's really 18 edges, 17 of the 18 then were either well known or confirmed by their experimentation. So that's about a 94 or 95% positive predictive value of precision. So that's pretty good. They did miss some. Those are shown with the dotted arcs. So in general, we wouldn't expect these methods to find everything that's causal, but what we're particularly interested in is them having a high positive predictive value, so that when they say there's an arc, there's a high likelihood it's there, and then the scientists can go find those that they don't currently know well, like the green ones, do experiments, and those experiments we would tend to be validate the purported causal relationships from the network analysis. So this I use in this illustration just because it was done with biomedical data, and it's small enough to sort of display on the screen what was going on. Some of the networks that we're dealing with now are much larger, but the same types of principles apply. So I'm going to sort of switch gears now and just talk for a few minutes about what are some of the basic components needed to learn these kind of causal networks from data. And there's really, you can break it down into 3 things, and I know that many of you may already know these things, but some of you may not as well, so I wanted to go over them in case it might be helpful. One is, of course, we need a model to represent causal relationships. And we need a way of searching over those models, because there are a lot of possibilities in general of what might be the causal relationships among the measured variables. And as we're searching, we need a way of evaluating these models so that we end up with a model that is the most probable, or a set of models that are all highly likely, sort of the top models. So let me go through this systematically with some simple examples. The representation that we're mostly focused on are called causal Bayesian networks. They're Bayesian networks where the arcs are interpreted causally in terms of direct causation. The nodes represent variables, the arcs represent, as I said, causation, and they're directed acyclic graphs. So they don't represent feedback per se, but there are temporal extensions that do allow representation of feedback, and there are extensions that under equilibrium conditions and linear Gaussian relationships can themselves be cyclic. The graphs themselves are cyclic. And basically a variable is modeled as independent of its non-effects, the things that it doesn't affect, given its causal parents. So, for example, here, a C is independent of A given its parents, B. And that's the basic notion here of if the only way for A to influence C is through B and you control B, let's say you fix B, then changing A will not influence C, change its distribution or its values. So that's the intuitive notion of the model. So this is called a causal Bayesian network structure. And then that structure factorizes the joint probability, factorizes to give a joint probability distribution. So in this example, what that would be is that we have a probability of A, because A doesn't have any parents, and we would specify that, a probability distribution of B given A, and a probability distribution of C given B, namely C given its parents B. And it turns out that you can show that the independence properties that you assume that I mentioned in the previous slide lead to this factorization, and therefore you can oftentimes represent the joint probability distribution with a compact number of parameters if nodes don't have a large number of parents. So it's a way of doing sort of more parsimoniously representing potentially complex joint probability distributions, big joint probability distributions with relatively parsimonious parameters. The next phase is that our next step is we also need to have model search on that representation. And so a issue is that the space of causal Bayesian networks is very large, and we generally have to use some sort of heuristic search to try to find the most likely networks. And once we find one, we can then parameterize it, and we can also model average over, let's say, the set of top networks to find relationships that are fairly consistent among those top networks. And we can quantify that in various ways to show the probability that there's an edge between, let's say, A and B, model averaged over the top few hundred networks that we find. So just to illustrate that with our simple three-node CBN, here's a possible kinds of networks that an algorithm might search over, looking for scoring these as it goes. And oftentimes this is a greedy search, basically searching through the network of possibilities in a greedy fashion, trying to find a local optimum. And then as it's searching, as I mentioned, we need to evaluate these networks. And there's two primary approaches, although there are certainly hybrids of this and other approaches that I don't have time to go into. But one is called constraint-based, which uses tests of conditional independence. And the other is Bayesian, or more generally score-based, where you're scoring a network in some sort of quantitative way. And a Bayesian approach is one way to sort of do that, where you're calculating essentially the probability of a network structure. And I'm going to focus on the Bayesian for the next few slides to show you how that could work. So here we have our causal model again, or at least one of them, with A causing B and B causing C, but A only influencing C through B. And we'll call that causal model structure S1. And then the bottom part of the slide is a set of parameters. And let's suppose that we're hypothesizing both the structure and this set of parameters. We'll relax this parameter setting assumption later. But right now, let's just suppose that we're actually going to score a causal Bayesian network that has both the structure and the parameters. And here you can see that the prior of A being true is 0.6. For example, the probability of B being true given A is true is 0.8, and C being true given B is true is 0.9. Now, in general, these could be more than binary, and also these could be continuous. And there's work going on where you have a mixture of these variables being discrete and continuous. But it's easiest to show it with just some binary variables, which is what I have here. But this table shows, for example, for B, every possible value of B given a value of A, and these are the conditional probabilities. So let's look and see, again, in a very simple fashion, how we would score if we had these four samples, these four cases that, in general, we would have hundreds or thousands, but we just have four here. So we're going to score each of these cases. What we would like to produce is essentially what is the probability of this data given this causal Bayesian network. For the first case where A is true and B is true and C is true, let's look and see what we would do. Well, the probability of A being true in this network is 0.6, and the probability of B being true given A is true is shown by here. That's 0.8, so that appears here. And finally, the probability of C being true given B is true is given by this conditional probability, which is 0.9, so that appears here. And then we take that product and we get 0.432. So the probability of this case, given this Bayesian network, is 0.432. If we do the similar type of thing for the next case, which is A is false, B is false, and C is true, we get 0.108. For the next case, true, false, false, 0.084. And for the final case, we get, again, 0.432, as we did at the first case, because it has the same values. If we assume these cases are IID, which is what we often do, and it's not a bad assumption oftentimes, sometimes it can be, we basically can take the product of these cases. They're independent given the model. They're not marginally independent, but they're independent given the model under IID. And that product is 1.7 times 10 to the minus 3 of the probability of this data given causal model 1. Okay? So this is another causal model that we're searching over. This one has A causing B, but in this case, rather than B causing C, we have C also having a causal influence on B. And there we might have that we have the same prior on A's values. We now have B in terms of both its parents. So B being true, given A is true and C is true, we might, for example, want to consider, hypothesize that that's 0.7 and so forth. And then C being true in this particular model, we're hypothesizing it's 0.6. So if we go through the same exercise on the same data that we saw in the previous slide, we will get the following. I'll just do the first case. So A being true is 0.6. B being true, now we have to look at both of its parents in this product decomposition. That's going to be A and C. So B being true, given A is true and C is true, is 0.7. So that appears here. And finally, C being true, according to this model, is 0.6. That's here. And the product gives us 0.25. And for the other three cases, we get these other probabilities for each of those cases. And when we take the product of the four, we get 8.8 times 10 to the minus 4 as the probability of the data given this causal model. Now what we do is we apply Bayes' rule. And so, for example, if we want to know the probability of causal model 1 given the data, we basically have already computed the probability of the data, the marginal likelihoods given causal model 1. And we need a prior for causal model 1. And we've also computed the probability of the data given causal model 2, which is shown here. And so this is just Bayes' rule. We have to have the prior of causal model 2. Let's suppose that our priors are 0.5, so we're sort of indifferent between these two networks a priori before the data. And so we put in 0.5 for each of them in terms of our prior belief that they're generating the data. We do this calculation. The 1.7 times 10 to the minus 3 is the one we got for the first network, and then the one we just got for the second is, as it's shown here, 8.8 times 10 to the minus 4. Anyway, we get 0.66 as the probability of causal model 1 given this data. And so this network, which is that causal model 1, would be, and its parameters would be more likely given those four cases. And in general, if we have a lot more cases, these probabilities can get much larger, closer to 1 or 0 for the various networks. More generally, what we do is we, this is kind of a generalization of what we saw before. We actually compute the structure, probability of the structures given the data. And the way we do that, because that's often interesting, what we're interested in is the qualitative relationships, and then we can parameterize those once we find a structure that's highly likely. So what we see here is that we integrate over the parameters. So this is the data given the structure and the parameters, and then we have a prior over the parameters given the structure, and oftentimes we assume something non-informative here. And then we have a prior of the structure as we saw before. But we integrate over this to get the marginal likelihood. And under certain parametric assumptions, we can get closed form solutions to these integrals in discrete and continuous cases. And so that's basically what this is showing. And then more generally, we don't just have two models, as we showed in the example, but in the denominator, we have to sum over many models. There could be thousands or hundreds of thousands of models that we're summing over, and we do this integral for each one. And that gives us the probability for the structure represented in the numerator over all those in the denominator, all the scores in the denominator. So that's the basic idea in sort of simple terms, a lot of details, but I hope that conveys some of what's going on. And really, it's it's fairly traditional statistical types of inference. It's just inference with causal models. We have a particular model that can be interpreted in a causal fashion that seems to have some reasonable semantic relation tie to causation in the world. Not perfect, but nonetheless some tie to causation in the world. And we use that model to try to estimate causal relationships. Now, I'm not able to go into a lot of details that you might be occurring to you, which is that so far we've just considered measured variables and not hidden variables. But one of the really difficult things about trying to discover causal relationships among from observational data is the potential presence of hidden confounders. That really causes a lot of problems, and one of the reasons why we do experiments to try to eliminate the influence of those hidden confounders. So this one at the top one just shows direct causation. The second row here shows that we have this H, which is an endogenous latent variable, that X is causing that, and then that variable, which we're not measuring, is causing Y and Z, which are measured. So X, Y, and Z, let's assume, are measured. Here's a simpler version, which is just latent confounding, where H is the latent confounder, and we've got X and Y being measured, and we find a statistical dependency between X and Y, and what we're interested in is it causal, or is it due to confounding, or maybe both? So we have to grapple with that. Another possible reason why X and Y might be statistically dependent is due to selection. So in this case, they're both influencing why we're measuring the data that we're measuring. So S could be essentially selection for having been sampled. And if you have these two variables influencing the sampling process, they can be statistically dependent for that reason. So we've got confounding, selection bias, and various combinations of the two and other things that can cause some difficulties. And algorithms have been developed to deal with these issues. Let me give you a few other issues or dimensions of the problem. We could have prior knowledge or not, deterministic or probabilistic. The variables can be continuous, discrete, or both. There can be temporal dynamics, stationary, non-stationary, continuous, discrete time, parametric or non-parametric distributions, linear or non-linear relationships, additive noise or non-additive, or possibilities. There could be feedback or no feedback. As I mentioned, latent confounders or not, selection bias or not. The data sets themselves might be single data set, but we also may have multiple data sets on the same variables, or multiple data sets on overlapping variables. So there's a lot of potential complexities, and we in the field of causal discovery and in the Center for Causal Discovery have addressed many of these in various ways individually, but I should say that there's still many open problems, and particularly open problems in trying to address them all simultaneously. So I don't want to leave you the impression that this is we're near the closure of having done what needs to be done to make this as useful as it can be, but we're making progress. We still have many open issues and opportunities to do better. So let me talk a little bit about big data. There's different dimensions of this. One way of cutting it is to look at the volume of data, the number of samples, the number of variables per samples are two different ways. There could be variety of data, different types of data. Velocity, how fast is that data coming at you, and how vast the uncertainty in the data can also be a form of complexity. So what are we doing in the CCD, Center for Causal Discovery? What is our focus in terms of big data? Well, Small problems have been, these methods have been applied to small problems, a small number of variables and samples, quite, reasonably successfully for a number of years. More recently, methods have also been successfully applied to medium-sized biomedical data sets where you may have hundreds of variables, for example, rather than just dozens. But most algorithms are not able to handle big data sets, by which I mean thousands, tens of thousands, or even hundreds of thousands of variables. And those kinds of data sets are becoming available now in biomedicine, particularly some of the omics type of data, such as whole genome data, gene expression data, and so forth. So let me illustrate that, the relevance of that in this table, where we're going to look at the number of causal models as a function of the number of measured variables. And I'm going to assume, just for simplicity, that we don't have any latent variables or no directed cycles. So if we have one node, obviously there's just one causal model. There's really not anything interesting to say causally. If we have two nodes, there's three models. A can be causing B, B can be causing A, or basically there can be no causal relationship between A and B. So those are the three. Again, I'm assuming no latents and so forth, no feedback. It would be worse if we did. If we go to 3, it's 25 possible models, 4, 543. If we get up to 10 nodes, I should say, we get up to over 10 to the 18th possible causal models. So, the space explodes, and we need some way to deal with it. So, the big data problem that we're focused on in the Center for Causal Discovery is to analyze biomedical data sets containing a very large number of variables to generate plausible hypotheses of the relationships that hold the causal relationships among those variables. So this is the number of variables per sample is what we're really focused on. So let me briefly mention some of the progress that we've made. We've optimized a popular causal network learning algorithm that's Bayesian, called GES, to develop a more efficient version, which we call FGS. The basic approach is we first optimized a single processor version, and then we parallelized it. And in a preliminary evaluation using simulated data, what we did was we generated a random causal Bayesian network containing 30,000 nodes and 60,000 edges. We had a continuous variable version, which used linear Gaussian relationships, and a discrete variable version, which uses multinomial relationships. And we sampled each CBN to generate 1,000 cases. And we provided those cases to the algorithm, FGS algorithm, and measured the performance to learn a CBN in terms of the precision and recall of finding causal arcs, and also the runtime. So this just shows the results of the first row is for the continuous variable version, and we actually generated data from 10 different networks. And so here we have 30,000 variables, 60,000 edges, and the arc precision was quite good, 99%. The average arc recall was around 84%. We used 128 processors on the Pittsburgh Supercomputing Center hardware, and it took about a little over 2 minutes to learn each network structure. With the discrete, the results were not quite as good. The arc precision was around 86%, and the arc recall was around 24%, and it again took around 2 minutes. So we want to be able to we're in the process of applying this to real data, of course, also, but these simulation experiments can be very helpful to give you a first cut idea at both the time complexity, runtime, and also the performance under different assumptions about how the data is being generated. And there's an archived paper by Joe Ramsey. So if you look at Ramsey, you'll probably should be able to find it shown here. Talks a lot more about this FGS algorithm and how it works and how it achieves its efficiency. And also Ramsey, Gleamore have a paper that's in I think it's in press called Million Variables and More that talks about this and some of its application to real data. So I wanted to just point out this FGS software is available from our website if you're interested. It's free and it's open source and it's on GitHub, so you can get at it through www.ccd.pitt.edu/tools, and we would be happy for you to use it. And if you would like our help in using it or have any questions, please let me know. I'll give you my email address. We have a software group that's able to help people that want our feedback, and we also have some tutorial material that you can find on our website. So the primary goals, just to sort of briefly review this, of the whole center are to develop and implement state-of-the-art methods for causal discovery from big biomedical data. And we want to make some of the best methods available in a free, open source, and very efficient fashion. And we want to develop new methods that are driven by feedback we get from solving real problems. A second goal is to investigate 3 problems within our center, and I'll talk briefly about them, to evaluate the usefulness of the methods and also to drive further development of them. And a third one is to disseminate the methods and knowledge widely to biomedical researchers and data scientists. So both in terms of algorithms and in terms of a desktop application. So those algorithms are available in R, Python, and in Java. A desktop application called Tetrad, which is a graphical user interface for using the algorithms. And we're collaborating with other centers. We have a summer workshop that lasts about a week, 5 days, at CMU. So if you're interested, that'll probably be in June, and you can look at our website to get more information once we figure out the exact dates, which we'll probably figure out in the early part of 2017, maybe January or February. But that gives basic training about both the methodology of graphical causal modeling and discovery and also some hands-on use and training of using the tools. And so we had one last June, and we also had a datathon. So we had 3 projects I mentioned. I'll just more or less list them. One is to discover cell signaling networks in cancer. And another is to discover the mechanisms of onset and progression of COPD and IPF to lung diseases. And a third is to discover the functional or causal connectivity in regions of the human brain from fMRI data. And I'll focus on this one for just a few minutes to give you a flavor for some of what we're doing with real data. So this is directed by Clark Gleamore in the center, and Clark also directs the algorithms group. He's at CMU, and he has a great group of people working with him on this project. The goal is to discover, as I mentioned, the functional connectivity of regions of the human brain using fMRI data, and they're interested in looking at differences of that connectivity for individuals that have autism spectrum disorder versus not having that disorder, schizophrenia versus individuals that don't have schizophrenia, and also just longitudinal studies of neurotypicals and in a resting state, and looking at those connections. So they had a poster that I'll just briefly summarize, which is titled Fusiform and Cerebellum fMRI Connectivity Implicated in ASD, Autism Spectrum Disorder, and you can see the authors of that paper here that was presented in the annual meeting of the Cognitive Neuroscience Society back in April. So an overview of the basic idea of their poster was that cellular, anatomical, and behavioral studies have found abnormal properties of the fusiform gyrus and the cerebellum in ASD. Some fMRI studies have reported contradictory results that did not support these conclusions, however. So there's some controversy. Whole brain functional correlation patterns have been used to classify ASD individuals in typical controls, but their accuracy hasn't been all that high, and I'll show you a table of that. It hasn't been terrible, but it hasn't been really high either. And so what this group did was estimate graphical causal models from just 10 ASD subjects, fMRI from 10 participants with ASD and 10 typical controls using a BI data set from Utah to show correct and complete measurements of the cerebellum. So those 10 actually showed very good measurements of the cerebellum, which is implicated as the earlier bullets showed, but there's some controversy about its role in ASD. So what they did was, within the center, was to use 10 edges from the graphical causal models that were found by their algorithms applied to this fMRI data on these 20 samples as features. These edges, causal edges that were found, were features in an SVM classifier, and they used that to then predict in a leave one out fashion, you know, ASD versus not. And they also just looked at the edges that involved the fusiform gyri and two regions of the cerebellum just to see how well they predicted, and I'll show you those results in a minute. So this is obviously a preliminary study, but it does sort of exercise a number of components of what the center is trying to explore. So those 4 edges actually did reasonably accurately in predicting, as I'll see, so I'm just giving you an overview. This is just, I'll have about another minute or two going into the details about their study, and then I'll finish this up. So again, 10 ASD and 10 controls. They did this various kind of data acquisition and pre-processing, time resolution of 2 seconds. They did various artifact removal and normalization, spatial normalization. They were using 2 millimeter voxels, and they did parcellation to about 100 regions of interest from the AAL atlas, and the regions of interest signals defined as average bowl signals over the voxels in those regions. And they applied FGS that we already talked about, which is a Bayesian method that we sort of illustrated with a simple example. A variation on the FGS algorithm they applied to their data. And they basically did do causal modeling, but then they just used the causal edges in the end to look and see to what extent they were predictive. And again, use support vector machines on those edges. And this shows, again, just the experimental flow, data pre-processing, FGS applied to causal models. They just basically looked at the edges that had some variance in them between the control and the ASD participants, and gave that to SVM and looked at accuracy of classification. So this is a transverse view of the brain, and it shows in color, well, some of the colors are black, but you can see these 10 edges and the red and the green. So the red are the ones that are prominent in the ASD individuals, and they're also involved the fusiform and the cerebellum, this back portion of the brain. And the green are the prominent in the controls. So they used all 10, and I leave one out to predict, and they actually got 100% accuracy with that. They used 4 edges, the ones in color, red and green, and they got 90% accuracy for that. So obviously these are very low numbers, and this needs to be repeated, but it's just giving you a sense of how some of this might be done with these algorithms on this kind of data. Other literature that is doing similar types of predictions of accuracy and being able to differentiate from fMRI data, ASD individuals from those without, show the accuracy here that's around the 70 or 80% range. So this does look like it was doing a bit better. The p-value on this was reasonably small. So in summary then of this study, all the other studies have larger abide samples than the 10, and they were focused on ones that had really good data on the cerebellum and fusiform gyri. And they did not include the control, the quality of those regions. And they're based on functional correlation patterns as opposed to doing a causal analysis of the essentially what causal effect is estimated to happen between one region and another. The sample was small, of course, but the connections, including the fusiform gyri of both hemispheres and 2 regions cerebellum, were pretty good accuracy, and so the results suggest that good measures of cerebellum regions do support accurate diagnosis of ASD. So what they're doing in this group within the center in the more recent past has been looking at resting state of one individual who's taken, I think, 50 or 60 fMRI measurements over time, separate, scans, sessions. This is Russell Poldrack at Stanford University. He's does work with fMRI there and is well known in this area. And they're basically doing causal inference at the voxel level. So there's about 50,000 voxels, and FGS is fast enough to be able to do causal discovery at the 50,000 voxel level. And they've been looking at that data that he's provided in collaboration and trying to understand the patterns in this data. So let me summarize my overall talk and then open it up for questions. Discovering causal relationships is at the core of much of biomedical science, and effective methods exist currently for representing and discovering causal relationships from data, including observational data, and particularly graphical models that we're focused on. And we're concentrated on developing and making available these algorithms and systems as open source code in various flavors available to the community for generating causal hypotheses from big biomedical data. And then we're focused on studying three particular biomedical problems as exemplars and drivers of these methods for causal modeling discovery in health and disease. And here's a few selected references. The first one just gives more of an overview of our center in more depth. The second one is an overview of automated search for causal relationships. It's in a book shown here. The third one is an article that's an overview of causal structure learning and inference by a group that is at ETH in Zurich, Bielman, and has been doing some very interesting work in that area. And then Pearl has a book that he came out with that's more focused on the modeling itself and the causal models, Bayesian networks, and what you can do with them, not so much on the discovery, but nonetheless a nice book, a primer on that aspect of causal graphical modeling that just came out this year. I wanted to thank the 60-plus members of the center for, for the work that I've been reviewing, and also thank the NIH for a grant on a BD2K grant that has supported this work. We're basically at the 2-year mark. It started in the fall of 2014, and this is a 4-year grant. So thank you. This is my email address here and the website for our center, and I'm happy to correspond with anyone that has would like to. And so now I'd be happy to open it up for questions, Sean, if that would be good.
Yeah, great. Thank you, Greg. Are there any questions out there? Some of you may be muted because during the presentation there was some background noise, so I muted you. So if you have a question, please unmute and speak up.
Laurent, can you hear me?
Yes, we can hear you, Laurent. Mm-hmm.
Okay. I was intrigued when you were talking about using experimental and observational data in the causal inference. Can you elaborate a bit more of the context within which you will be, how would you use differently the experimental and the observational data, and do they have to be done on the same, the units being the same, a measure on the same variable, just so that I understand a bit more?
Well, that's an interesting question. I mean, a lot of times, when the experiments are done biologically, as you know, you're manipulating a small set of variables and then measuring many more. Particularly as we get into sort of doing sort of high throughput experimentation, we're oftentimes manipulating the system in a few ways, but then we're measuring a whole lot of other things. So I think the center of mass is probably the most common case is probably going to be and is going to be a combination of the two. Some of the variables, a few being manipulated and others being perturbed and others being measured. So I think that Karen Sachs' paper that I used as an exemplar really is nice in that respect because it shows that being done on some real data. And I think that to answer your other question, technical question, the Bayesian methods, which is what she used, have a nice property that they are able to take, every case could be manipulated in a different way with some overlap. But you basically can have an arbitrary set of variables being manipulated, and they could be different from one case, one sample to the next, and the Bayesian methods can integrate over that, can deal with that. The constraint-based methods, it's more problematic. Those can deal with if you have a data set that has a variable that has been manipulated, then you're fine, or a set of variables and consistently manipulated over the entire set of samples. But it's more problematic to deal with changes in manipulation from one sample to the next, or to deal with, let's say, 5 subsets of samples that one subset is manipulated in one way, another subset is manipulated in a second way, and so forth. So does that answer your question?
Yes, it does. I was asking the question in the context of discussion with RCT for pharmaceutical products, for instance, where discussion were about the need to keep context information because the intervention may have worked in one deployment condition and not in the other. So what you are saying is that in some way those observed variables are used to look at context within which your expected causal links are working and not working. Am I correct in my yeah? Okay.
Yeah, I mean, I think I was probably answering part of something you didn't ask, but your point is yes, I agree. We can have variables, and oftentimes we need to have variables that provide some information about the context in which the other variables are being measured.
Okay, that's good.
Yeah.
Okay, great. Any other questions?
I have another one, but I don't want to take. Okay. My other question is with respect to the velocity aspects, which I found very interesting within the context of I was telling you that I am in a business school, and a lot of the big data would be used, for instance, to really have ongoing monitoring and then providing real-time feedback to individuals as a function of various contexts within which they may be. So I would like you to talk a bit more about the velocity challenge and then the link with the context where it's not the velocity of acquisition of information, but also velocity at which you need to feed this back to a type of user.
Well, yeah, I mean, there's a whole bunch of interesting questions there, and I think as we start getting into the mobile technology and start measuring more and more regarding individuals in the context in which they're living, there's going to be a lot of velocity of information about individuals and about groups of individuals, which is, I think, going to be challenging. I think, so, right now where we are, in my opinion, is, at least in biomedicine and in clinical healthcare, we, the velocity is not challenge, and so that's why we haven't concentrated on it, focused on it. We think that just the number of sort of variables, you get a lot of variables that are being measured now, but they're only being measured maybe once or a few times. So velocity is not the dominating issue there. It's really the quantity of variables. And that's because of the combinatorics of the variables in terms of teasing out causation. So I think that basically, to summarize, what I'm saying is that I think, in my opinion, at least within the biomedical data that I know, velocity is not the main problem, but I think velocity is going to become increasingly more relevant as we start getting real-time systems that are measuring what's going on, continuously and in real time. So it will become an increasing challenge.
Are the constraints primarily at the level of computational time, or are they of another nature?
Well, I was thinking in terms of just computation time. There may be other dimensions too, but yeah, I was focused on computation time.
Yeah, the reason, still to give you the context within which I was asking this question, we work on the system dynamics side with colleagues from MIT, and if you look, John Sermon, who has been one of the pioneers in terms of very complicated model of system dynamics of climate change and so on. And in order to use assemble those variety of models to be able to be providing feedback in real time, for instance, at the UN meetings, because of computational velocity constraint, having to develop kind of a synthetic version of a different model and get them. So that's why I was asking this question to what. So that's very useful.
Yeah, well, and it's related to a topic that I didn't get a chance to go into, but we've been exploring it in the cancer project within the center, and that is to do personalized modeling, precision medicine, where we're trying to model what is going on in an individual, in this case, an individual tumor, and trying to understand the causal relationships in that tumor, which could be quite different than the causal relationships in the same tumor type or even subtype of another patient. And so when you get into doing personalized types of causal modeling, you get into, in some ways, a lot more challenges in terms of trying to build those models for each person, because you can't just build one model for the population and kind of use that on many people. You have to actually do search and discover models for each person. And so there's some additional computational challenges there as you start getting more information coming in about these people, maybe gene expression levels over different tissue types that are coming in. You need to update your personal models for those individuals, and so you may get into some computational challenges for that. If you're doing that for millions of people, or thousands of patients in a hospital system. So I think this is in the future, but I can certainly appreciate that it's likely to be coming.
Am I allowed for a last question, Sean?
Let me just check to make sure. Does anybody else have a question? Please go ahead, Lorette.
Okay. So my question is with respect to the actual quality of the prior knowledge. The bulk of this modeling has been done within the biological space or the medical space, where there is 300 years and more of research for various causal links. And I was telling you at the beginning that we want to learn from this to try to examine human with their biology within the social and economic context. So my question to you is pragmatically, what are the possibility of validly using those approach within the context of social, economic, and so on, where the causality between variables, the scientific background is much less complete than in the biomedical side. And my question beyond this is, how good has to be the evidence for the causal link to have a model that is actually having solid validity?
Well, I mean, I think if you don't have as much prior knowledge, puts more of an onus on the data for causal discovery, and then it boils down to do you have the kind of data that represents the variables that you're interested in, that's relatively well representative of what's going on, and do you have a reasonable number of samples? So, I mean, I know I'm doing a lot of hand-waving here, but it depends on the details.
Mm-hmm.
But these algorithms can run without prior knowledge. So, in effect, what you're doing is sort of, in the Bayesian framework, you're giving effectively a non-informative prior over the structure and parameters. And you can do that. And actually, a lot of times, these algorithms are run in that way on data sets to see what you get. So, you don't have to have a lot of knowledge to run them. But how good the results are is going to depend, not surprisingly, on how good your data is and how much data you have.
Okay, thank you.
Sure.
Okay, well, Greg, thank you very much for a beautiful presentation and joining us today to let us know about your center and the research. And I just remind everybody that next week we'll have Professor Franks to give us a webinar, and we hope to see you there. Thank you very much, everybody.
Thank you. Thank you, Greg. Bye-bye.
Thank you.